Today we are going to discuss about the chromosomal structure and histones. Chromosomes are thread-like structures appear at the time of cell division in the nucleus. The following slide shows the detailed structure of the chromosomal condensation or hierarchical condensation, where the DNA is going to form the nucleosome model, where the histones are going to take part to form a nucleosome model, where the nucleosome model will form the scaffold model, and the scaffold model will give rise to the chromosome structures. Chemical composition of chromosome is DNA, protein, and a significant amount of RNA are the sites of the RNA synthesis, where the DNA is going to comprise of 40% of the chromosomes and the proteins are going to comprise of 60% of the chromosomes. Eukaryotic cell stores its genetic information in DNA molecules that can be over 1 meter in length. So if you are going to stretch the DNA of the eukaryotic cell nucleus, it is going to have the length of 1 meter in length. The following slide shows how the DNA has been formed the scaffold model, and further the scaffold model is going to form the chromosome thread-like structure. The next slide. The DNA is hierarchically packed in the nucleus up to 2 into 10 to the power of minus 5 times smaller in length, with the aid of proteins to form a complex called chromatin. Proteins are the histones. Histones, proteins that DNA tightly coils around to form the chromosomes. A group of 5 small basic proteins occurring in the nucleus of eukaryotic cells that organize DNA strands into nucleosomes by forming molecular complexes around which the DNA winds. Nucleosome core particle organization will be seen further. The nucleosome core particle represents the first level of chromatin organization. Under this, histones are the proteins associated. This is the structure, how the nucleosome is going to form out of the proteins, where whatever the H2A, H2B, H3 and H4 you are seeing, those are the core particles of where the 4 histones has been shown in this picture. Nucleosomes composed of 2 copies of each of histones, H2A, H2B, H3 and H4, assembled in an octameric core with 146 or 147 base pairs of DNA tightly wrapped around it. These octamer are about 147 base pairs of DNA wrapped in 1.7 spherical turns. Nucleosomes are connected by a DNA linker of variable length that forms a 10 nanometer beads on a string array. Nucleosome cores are separated by linker DNA of variable length and are associated with the linker histone H1. In this you can see about 6 nucleosomes where the DNA has been 1.7 spherical round has been seen, H1 histone has been seen, which has been attached to the DNA, which is the linker DNA. Octamers is also been seen. The next level of chromatin organization is the 30 nanometer fiber, which is composed of packed nucleosome arrays are found to be arranged as a two-start helical model and mediated by core histone internucleosomal interactions. This is the 30 nanometer structure where the octameric histone core molecules are getting condensed further to give rise to a scaffold model of 30 nanometer. Histone structure and nucleosome stability will be discussed now. This is the scaffold model, which again describing the DNA nucleosome scaffold. Histone proteins play essential structural and functional roles in the transition between active and inactive chromatin states. Active chromatin states are the gene expression chromatins, and inactive chromatin states are the gene silencing or non-expressing regions, or the non-expressing state of the DNA. Histones have a high degree of conservation due to constants to maintain the overall structure of the nucleosomal octameric core. The evolution is very less in the histone proteins, but variants have evolved to assume diverse roles in gene regulation and epigenetic silencing. A few variations has been observed to assume diverse roles in gene regulation and epigenetic silencing. Overall structural state of chromatin and its domains controls both DNA replication and the expression of genes. Structural chromatin have been implicated in transcriptional activation, silencing chromatin assembly, and DNA replication. Epigenetic modifications are switches for the regulation of gene expression and are chemical modifications of the DNA and histones that do not result in changes to the DNA sequence. Thank you for watching this video. Please subscribe to our channel by pressing the button below.